Imagine trying to find a single, specific fingerprint. Not in a room, but on a suitcase that's currently somewhere in the entire global air freight system.
That's a great analogy.
It just seems impossible, right? But that's pretty much the challenge doctors are up against when they're hunting for the absolute earliest signs of disease.
Exactly. We're talking about biomarkers, these tiny little signals the body gives off, maybe a single protein from a cancer cell that's just starting out, or, the first whisper of neurodegeneration, years before you'd ever notice anything.
And right now, our best tools, they're just not good enough for that, are they?
They're good, but not that good. The gold standard is something called ELISA. It's a workhorse, but it can only detect things down to the picomolar range.
Picomolar sounds tiny, but in this world, it's actually pretty loud.
It's millions of molecules, which means you're usually finding the disease only after it's already, set up shop and is flooding the system with these markers.
We've got fancier stuff, of course. Digital ELISA can literally count single proteins, but that's a huge, expensive lab machine. The turnaround time isn't great.
And that's the whole problem. For real preventative medicine, you need something that's faster, something way, way more sensitive, and ideally something portable.
And here's the crazy part, the paradox we're diving into today. That technology, we already have it.
We do. And get this, it's been around for more than 35 years. They're called FETs, field effect transistors. And they have shown detection limits that are they're almost unbelievable. We're talking down to the 10 zeptomolar range.
Hold on, zeptomolar, I don't even know what that means. Put 10 zeptomolar in perspective for it.
Okay, so 10 zeptomolar is basically the physical limit of detection. If you have a normal-sized drop of blood, say 0.1 milliliters, you are detecting maybe one. A single target molecule.
One molecule.
When you're finding a single ghost in the entire chemical machine.
So if we have a sensor that can find one single cancer marker, blowing every other tool out of the water, why isn't it in every doctor's office? Why have I never heard of it?
That is the big question. And it's the mission for this deep dive. We're leaning on this fantastic perspective piece by Eleonora Macchia and Luisa Torsi from the Università degli Studi di Bari Aldo Moro and Albo Academy University.
And what did they find?
Well, their paper in Nature Reviews Materials really cracked the code. They figured out what makes these sensors so incredibly powerful and also what makes them fail commercially. It's this amazing, almost accidental science.
All right, I'm hooked. Let's start with the basics. What even is a field effect transistor, an FET?
Think of it like a dimmer switch for electricity on a tiny, tiny scale. You have a semiconductor channel where current flows from a source to a drain.
Okay.
And you control that flow with a little knob called a gate. Applying a tiny voltage to that gate acts like a valve. You can open it up, let more current through, or choke it off.
So it's like electronic plumbing.
Yeah.
The gate voltage is the handle on the faucet.
Perfect analogy. Now, to make it a biosensor, you take that gate, that control surface, and you cover it with a super dense layer of antibodies. I mean, trillions of them.
An entire army of molecular detectors.
A ridiculously packed army. So now, when your target, the disease marker, comes along and binds to one of those antibodies.
That binding messes with the electricity on the gate?
Exactly. It causes a tiny electrostatic shift, a little electrical disturbance, right at the gate surface. And the transistor is built to sense that. It amplifies that tiny event into a big, measurable change in the current flowing through the channel.
And because these are chip-based, you can make them small, portable.
You got it. That's the dream of point-of-care diagnostics. A result in minutes from a little handheld device.
But Macchia and Torsi, the authors, they say it's not that simple. The performance isn't uniform. They talk about two totally different sensing regimes.
Yes. Let's start with regime A. This is the simple one, the predictable one. They call it the double-layer regime. It only works at high concentrations.
What's the mechanism here?
Yeah. Well, it's just mass stacking.
Mass stacking, like just piling things up.
Pretty much, yeah. At high concentrations, you have so many of the target molecules that they don't just find an empty antibody to bind to. They form a whole second dense layer right on top of the first.
So you're just measuring the physical weight, the electrical effect of all that stuff, piled on the sensor.
It's a brute force signal, and it's reliable. We see it in devices that detect things like C-reactive protein, a common inflammation marker. It works just fine in that high concentration range, but it doesn't explain the zeptomolar magic.
Right. Which brings us to regime B, the delta pH-enabled regime. This is where things get weird. How can one single molecule possibly create a signal that anyone can detect?
This is the wow fact of the whole story.
Yeah.
It's what governs that ultra-low detection. The authors use this incredible analogy. They say detecting one molecule is like spotting a single droplet falling into a lake 1 square kilometer in size.
You'd never see it.
You would only see it if the ripples were somehow massively amplified.
So what's the amplifier?
It's a kind of biochemical amplification. And here's the craziest part. It's enabled by something that scientists were often doing completely by accident. A tiny change in pH.
Wait, an accident? An accident is the key to the world's most sensitive sensor.
It seems so. So the antibody and the antigen, they bind together in a solution like blood or saliva, which has a stable physiological pH.
Makes sense.
But then, to get the best electrical reading, the researchers would often wash the sensor or move it to a different solution,
Yeah.
a low-salt buffer. And that buffer often had a slightly different pH.
So they're giving the sensor a little chemical shock.
A tiny shock. But that tiny pH shift, that delta pH, it causes these irreversible changes in that super-packed layer of antibodies. Think of them all, just slightly unfolding. It conditions them.
So the whole surface is now on a knife's edge, electrically speaking.
Precisely.
Hmm.
And now, when that one single target molecule finally finds an antibody to bind to, boom, that one binding event triggers this huge conformational change in that one antibody. And because they're all packed in so tightly, that change doesn't stay local. It propagates.
Like a line of dominoes.
The perfect way to think about it. One domino falls, and it electrostatically shifts the potential of millions of its neighbors. It turns a tiny, invisible ripple into a tidal wave of a signal.
That is just astonishing. Do we know for sure this is what's happening?
The evidence is pretty strong. The
Yeah.
paper notes that if you are extremely careful, if you keep the pH exactly the same when you switch to the measuring solution, the effect vanishes.
Wow.
The subatomic sensing is just gone. The amplification requires that pH shock,
Yeah.
and the results, when it works, are just stunning. A graphene FET detected SARS-CoV-2 antibodies down to 2.6 attomolar, that's maybe 150 molecules, and it did it in 2 minutes.
2 minutes. That's a game changer for screening.
Or an IgZO FET for a bladder cancer marker. It got down to 2.7 attomolar, which is something like 49 molecules in a tiny drop of urine.
Okay, so we have this sensor that operates at the literal limit of physics, and we now understand the accidental chemistry that makes it work. So I have to ask again, what is the fatal flaw? Why is this still stuck in the lab?
The core limitation, a thing that has stopped it for 35 years, is the problem of quantification.
Meaning you can't get a reliable number out of it.
Exactly. You can't use it for precise quantitative analysis.
But wait a minute, the signal, the voltage shift, it clearly changes with concentration, right?
Uh-huh.
You add more stuff, you get a bigger signal. Can't you just draw a curve and calibrate it?
You'd think so. It even looks like a perfect logarithmic curve, just like the Nernst equation that governs other sensors, like a standard pH meter. But the physics are totally different.
How so?
With a pH meter, you're measuring the actual movement of integer charges hydrogen ions. That's a direct, predictable process. It's stable. It's quantitative.
And with an immunoassay, you're not measuring moving charges?
No, you're measuring these subtle net neutral dipolar charge rearrangements. You're measuring the electrical effect of a protein changing its shape, its orientation.
That sounds way less stable.
It is. It's like trying to measure the mood of a crowd instead of just counting the people in it. The exact signal can be affected by the batch of antibodies, by temperature, by the specific way that pH shift happened, so you can't create a universal calibration.
So you can't say, this patient has exactly 2.7 attomolar of this marker, which means stage 1 cancer.
You can't. And if you can't trust the number, you can't use it to stage a disease or track how a treatment is working. That quantification failure is the roadblock.
But it seems like there's a pivot happening, a way to reframe the problem.
There is, and the authors highlight this brilliant example, a platform called SYNOT, which stands for Single Molecule with a Large Transistor.
And what did they do?
They just sidestepped the problem entirely.
Well,
They decided to stop trying to make the sensor something it's not. They chose to treat it purely as a qualitative binary test.
not how much is there.
But simply, is there at least one molecule present? Yes or no?
That's genius. You leverage its insane sensitivity for what it's good at. A simple, high-confidence yes or no for screening.
And it worked incredibly well. In trials for detecting SARS-CoV-2 in saliva, they had less than a 4% error rate for false positives or negatives. It proves the immediate commercial path for this is not as a lab quantification tool.
But as the most sensitive early warning system ever created.
Exactly. It's not about fixing the flaw, it's about redesigning the question you're asking it to solve.
This has been such a fascinating deep dive. Okay, let's try to boil it all down. What are the key takeaways here?
Okay, first, FETs are analytically the most sensitive biosensors we have ever created, period.
Yeah.
They can detect single molecules, which is far beyond our conventional tools.
Second, that incredible sensitivity isn't magic. It's actually this wild, often accidental process of biochemical amplification. A little pH shock conditions the surface, so a single binding event sets off a massive, measurable chain reaction.
And third, the big roadblock has always been that they aren't quantitative. They measure protein shape-shifting, not stable charges. So their future, at least their immediate future, is as ultra-fast, ultra-sensitive, qualitative screening tools. A simple yes or no.
The big picture here seems to be that the gap between the lab and the clinic wasn't a technology problem, it was a conceptual problem. We were asking the wrong question. To finally bridge that gap, we need to embrace what it's good at.
We have to stop trying to measure the mood of the crowd and just ask, is the crowd here?
So that leaves me with a thought-provoking question for you, our listener, to mull over. If a simple, cheap, portable device could tell you with near-perfect certainty that there is one single cancer precursor molecule in your system, even if it can't tell you the exact concentration, is that one qualitative yes enough to completely change the face of preventative medicine?
Something to think about for sure.
Thanks for diving deep with us into the 10 septimolar secret. Make sure you subscribe to the Deep Dive channel so you don't miss our next analysis.